Have you ever wondered how your body grows, heals wounds, and even creates new life? The answer lies in one of the most fundamental biological processes, cell cycle and cell division. This topic is crucial for NEET, so stay with me till the end for a clear and detailed breakdown. What is the cell cycle? The cell cycle is a series of events that allow a cell to grow, prepare for division, and eventually split into two daughter cells. It plays a vital role in growth of an organism, tissue repair, reproduction. The cell cycle consists of two main phases, interphase. 1, the preparation phase, where the cell grows and gets ready for division. 2, M phase, mitotic phase, the actual division phase. Now let's explore each phase in detail. Interphase, more than just resting. Although it's called the resting phase, the cell is actually very active during interphase. It is divided into 3 stages. G1 phase, the growth phase. The cell increases in size, synthesizes proteins, and produces organelles. mRNA and enzymes required for DNA replication are synthesized. The duration of this phase varies depending on the cell type. Example, liver cells take longer in G1 than intestinal cells. Checkpoint. Before moving forward, the cell ensures conditions are favorable for DNA replication. If not, it enters the G0 phase, a resting state. S phase, synthesis phase, DNA replication begins. DNA replication occurs, doubling the DNA content, 2N4N, while keeping the chromosome number the same. Each chromosome duplicates into 2 sister chromatids. Histone proteins are synthesized to help pack the DNA properly. Example, in humans, the DNA content increases from 2C to 4C, but the chromosome count remains 46. Why is this important? Any error in DNA replication can lead to mutations and genetic disorders. G2 phase, the final checkpoint. The cell prepares for division by producing tubulin proteins, needed for spindle formation. Organelles like mitochondria and Golgi bodies duplicate. The cell checks for errors in DNA replication before division. Checkpoint. If errors are found, the cell either repairs them or undergoes apoptosis, programmed cell death. Think of it like revising before an exam. If you're not fully prepared, you won't pass. Emphasis. The division begins. After interphase, the cell enters M phase, where it actually divides. Mitosis occurs in 4 stages. 1. Prophase, the setup stage. Chromatin condenses into visible chromosomes. Centrioles move to opposite poles in animal cells. Spindle fibers start forming. The nuclear membrane breaks down. Example, in human cells, each chromosome now has 2 chromatids attached at the centromere. 2. Metaphase. Chromosomes line up at the center, metaphase plate. Spindle fibers attach to kinetochores of chromosomes. Neat fact, metaphase is the best phase to study chromosome structure under a microscope. 3. Anaphase, the separation stage. Spindle fibers pull sister chromatids apart toward opposite poles. The centromeres split, ensuring each new cell will get a complete set of chromosomes. Neat tip, for a brief moment, the chromosome count doubles before cytokinesis occurs. 4. Telophase, the final step. Chromosomes decondense back into chromatin. The nuclear membrane reforms around each set of chromosomes. Spindle fibers disappear. Think of it like finishing an exam and neatly packing your notes back into your bag. Cytokinesis, the actual division. This is the final step, where the cytoplasm divides to form 2 identical daughter cells. In animal cells, a cleavage furrow forms and pinches the cell into 2. In plant cells, a cell plate forms due to the rigid cell wall. Final check. Each daughter cell gets exactly the same genetic material as the parent cell. Now that we've understood mitosis, let's explore meiosis, a process essential for reproduction and genetic variation. Meiosis occurs in germ cells, sperms and eggs, and reduces chromosome number by half. It consists of 2 divisions, meiosis 1, reductional division, and meiosis 2, equational division. The result? 4 genetically unique haploid cells, N, from a single diploid cell, 2N. Now let's break it down step by step. Meiosis 1, reduction division. The goal of meiosis 1 is to separate homologous chromosomes, reducing the chromosome number by half. It consists of 4 stages. 1. Prophase 1, the longest and most important phase. Chromosomes condense, become visible, and pair up as homologous chromosomes, one from each parent. Homologous chromosomes undergo synapsis, pairing, and form a tetrad, 4 chromatids. Most importantly, crossing over occurs at chiasmata, exchanging genetic material between non-sister chromatids. Nuclear membrane dissolves, spindle fibers form. Why is this important? Crossing over creates genetic variation, ensuring that no 2 gametes are identical. Errors in crossing over can lead to genetic disorders like Down syndrome. Neat tip: crossing over only occurs in meiosis, not mitosis. Metaphase 1, the chromosome alignment. Homologous chromosome pairs line up randomly at the metaphase plate. This randomness is called independent assortment, which increases genetic variation. Spindle fibers attach to centromeres of homologous chromosomes. Neat fact: each gamete gets a unique combination of maternal and paternal chromosomes. 3. Anaphase 1, the separation begins. Homologous chromosomes separate and move to opposite poles. Unlike mitosis, sister chromatids remain attached at the centromere. The chromosome number is now halved, haploid N. Important: if chromosomes fail to separate properly, it leads to non-disjunction, causing disorders like Turner syndrome or Klinefelter syndrome. 4. Telophase 1 and cytokinesis. 2 cells are formed. Chromosomes reach opposite poles. Nuclear membrane may reform. Cytokinesis occurs, forming 2 haploid daughter cells, N, but with replicated chromosomes. Key point: these cells are genetically different due to crossing over and independent assortment. Meiosis II, equational division. Meiosis II is similar to mitosis, but it separates sister chromatids instead of homologous chromosomes. No further DNA replication occurs. 1. Prophase 2, the setup phase. Chromosomes condense again. Spindle fibers form. Nuclear membrane dissolves, if it had reformed, in telophase 1. Quick note: no crossing over occurs here. 2. Metaphase 2, chromosome alignment again. Chromosomes line up at the equator, just like mitosis. Spindle fibers attach to the kinetochores of sister chromatids. 3. Anaphase 2, the final separation. Sister chromatids separate and move to opposite poles. The chromosome number remains the same, NN. Neat tip: errors in this phase can cause aneuploidy, leading to conditions like Down syndrome, trisomy 21. 4. Telophase 2 and cytokinesis. 4 unique cells are born. Chromosomes decondense back into chromatin. Nuclear membrane reforms. Cytokinesis divides the cytoplasm, forming 4 genetically unique haploid cells, N. Final outcome: each gamete has 23 chromosomes in humans, N = 23. When sperm N fuses with N, a zygote, 2N = 46, is formed, restoring diploidy. Imagine meiosis as a lottery. Each gamete is unique, increasing genetic diversity in a population. Let's quickly recap what we learned. Mitosis produces 2 identical diploid cells, 2N, used for growth and repair. Meiosis produces 4 unique haploid cells, N, used for sexual reproduction. Key features of meiosis: crossing over, prophase 1, genetic variation, independent assortment, metaphase 1, unique gametes, reduction division, meiosis 1, halves chromosome number, equational division, meiosis 2, separates chromatids. Why does this matter for NEET? Questions often ask about crossing over, chromosome count, and disorders caused by errors in meiosis. Meiosis is crucial for reproduction, evolution, and genetic variation. If this helped you, like, share, and check out my NEET Biology notes for quick revision. See you in the next video.